The American Brain Tumor Association is pleased to welcome you back to our webinar series. Our webinar topic today is Understanding Your Diagnosis Report. Thank you to today's webinar sponsor, GT Medical Technologies, for their support of this program. My name is Umbreen Maan, Program Manager here at the ABTA, and I'm delighted to introduce today's speaker. Dr. Erik Sulman is the co-director of the Brain and Spine Tumor Center at NYU Langone's Perlmutter Cancer Center. He treats patients who have brain and spinal cord tumors using radiation therapy, including gamma knife radiosurgery and external beam radiation therapy. Dr. Sulman is involved at the national and international level in current clinical trials and research. His research focuses on finding new treatments, particularly those that improve the effectiveness of radiation therapy, and on identifying molecular predictors of treatment response. Thank you so much for joining us, Dr. Sulman. You may now begin your presentation.
Thank you, Umbreen, and I want to thank the ABTA for the privilege to really speak to all of you who have joined us this afternoon, and I hope to give you some new information or perhaps clarify some things for you. The time is limited, so this will not be every possible new development in the treatment of malignant brain tumors. But hopefully it will provide some new information that you will find valuable. Briefly, these are my disclosures, that is specifically entities that I have either research funding or advisory support from. So again, I wanted to welcome you, and I'm going to just give you a quick overview about what I will discuss today. So when we talk about malignant brain tumors, we're really talking both about primary brain tumors, those tumors that originate within the brain or the spinal cord, and secondary or metastatic brain tumors. Those are tumors that come from other cancers elsewhere in the body that spread to the brain. I'll talk about some recent and ongoing clinical trials, and then I'll touch on some new technologies that may be having an impact in the future. And then, of course, I'll try to answer some questions through the Q&A. So I want to emphasize that the goals of this are really to talk about some of the latest developments for patients and to provide some tools that really will help patients and caregivers deal with the very complex decision-making that occurs from diagnosis through all the different treatments that you're challenged with. And then I'll give a preview of some of the new things that are coming down the line. And again, I want to emphasize that it's not possible to talk about every possible malignant brain tumor, every clinical trial that's out there, or every new technology. There just simply is not time to do that. But this will at least give you some of the tools that may be helpful in your discussions with your care providers. So let's start with primary brain tumors. As I said, these are tumors that originate in the brain tissue itself. Often we refer to these primarily as infiltrating gliomas, and I think many of you have heard the term infiltrating, which means that cells from these tumors go individually into the normal brain tissue. Now, as you can see from this pie chart in red, the most common of the malignant primary brain tumors is glioblastoma, accounting for almost half of them. And then the other tumors similar to glioblastoma, the anaplastic astrocytomas and lower grade astrocytomas, account for another third or another 17%. So roughly 67% or 65% of tumors are of those two varieties, which are sometimes called astrocytic tumors. Sometimes we also talk about meningiomas. Meningiomas are the most common tumor inside the head. They're not actually in the brain tissue, but in the lining of the brain. Of the meningiomas, however, only a small percentage are malignant. And that's why in this pie chart they have a very small percentage. But since they are very common tumors in the skull, that I will talk about them to some extent over the next few minutes. Another important thing to note is that primary brain tumors very, very rarely spread outside of the brain. And for that reason, we don't use the term metastasis to refer to primary brain tumors because they really don't spread away from the brain. And we also don't talk about staging when we talk about primary brain tumors, because staging is something used in cancer to refer to the degree that a cancer has spread to other areas of the body different from where it originally started. So the first thing I want to bring to all of your attention is that there's been an update in the way we classify and diagnose brain tumors. So, and this is primarily for primary brain tumors. The World Health Organization, often just abbreviated as the WHO, classifies all tumors of the central nervous system. And they've recently released and finalized a fifth edition. It was supposed to come out in 2021 because of the pandemic. It's coming out this year. The actual printed copy is not yet available, but will be shortly. But I think it's worth noting what are the big changes to the WHO classification, because this will influence the discussions you have. So the big thing, and many of you may know about isocitrate dehydrogenase, or the IDH gene. This is a gene that has really changed the way we think of infiltrating gliomas. And we can now very clearly divide tumors into either IDH, or isocitrate dehydrogenase mutant, or non-mutant. Any tumor that does not have a mutation in isocitrate dehydrogenase that is considered a infiltrating glioma, and also has certain other molecular changes, such as gain or extra copies of areas of chromosome 7, particularly the area that encompasses the epidermal growth factor receptor gene, which you may have heard of abbreviated as EGFR, or has a loss in areas of chromosome 10, which often harbors a gene called P10, not related to the number 10, and or has mutations in the regulatory region, the region of the gene that controls how the gene is turned on and off, of a gene called human telomerase. Human telomerase is a protein responsible for maintaining the ends of chromosomes. As cells divide, particularly in cancers, the ends of chromosomes wear down, and unless they're regenerated, the cell will actually die. And so cancer cells have adapted ways to regenerate the ends of chromosomes, and one of them is mutation in the promoter or the regulatory region of human telomerase. So if any of those three alterations are present, and there is no mutation of isocitrate dehydrogenase, we now call those glioblastoma, even if under the microscope they don't fulfill the old criteria for glioblastoma. So this then expands what we call glioblastoma. If there is an IDH mutation in the tumor, it cannot be called glioblastoma, regardless of how it looks under the microscope. And so that's another big change. And if the tumor has no mutation of IDH, so it's without an IDH mutation, so-called wild type, and it doesn't have one of those three alterations I mentioned just a moment ago, then it wouldn't be called a glioblastoma, but is one of these other entities I've listed here. Diffuse midline glioma that may harbor another mutation called the H3K27 mutation, a diffuse hemispheric glioma called an H3G34 with an H3G34 alteration. And if those two alterations aren't present, we actually call it a diffuse pediatric type high-grade glioma with both the H3 and IDH genes being normal or wild type. And even so it's pediatric type, those types of tumors can rarely occur in adults. So that was a somewhat detailed explanation, but it just shows that the definitions of what is a glioblastoma and what is not a glioblastoma have in fact changed with this new classification. One of the small changes, which you may see if you ever see a pathology report, is that we no longer call things grade with 1, 2, 3, or 4 using Roman numerals. We now have switched to Arabic numerals, obviously a much smaller change. And this is just showing you a diagram for the infiltrating gliomas, how things have now been reclassified. And what you see is that there's a group that is now what we call molecular GBM, where they are IDH wild type, but which have one of these three alterations making them molecular GBM, even if they would not fall under the glioblastoma criteria under the microscope. Okay, so what does it mean with this new World Health Organization classification system? How does that impact patients and their care? So we now use what we call an integrative diagnostic criteria. So we use how things look under the microscope, but as I said, we also use these molecular features, things like mutations, genetic changes. This actually complicates things, and in terms of interpreting the results of past clinical trials, because past clinical trials did not include this information. We simply didn't know about some of these genetic changes, and so when patients were included on those trials, we included patients that were a mixture of some having the genetic changes and some not. So as a result, it makes recommending treatment somewhat complicated because we don't always have the complete picture, and we can't go back in time and sort of reanalyze all of the clinical trials that have been done so far using the modern updated criteria. For one thing, we don't have the information on many of the patients in those clinical trials. We may not have collected the tumors for those trials, or we may only have a portion of the tumors collected that can be analyzed now looking back. And the other challenge, of course, is that there's a statistical problem. When we design a clinical trial, we design it with a certain set of assumptions and understanding about who will be enrolled. And if we now realize that a subgroup of patients should not have been enrolled in that trial, or maybe were imbalanced between the two treatment groups in that trial, it's hard to go back and reanalyze the data. I mentioned that we don't use staging any longer. We don't typically use staging to describe primary brain tumors. There is a staging manual that has been used by oncologists for many, many years called the American Joint Committee on Cancer Staging Manual. And the good news is this has not been very relevant for primary brain tumors, but is now being updated to include all the information I just showed you. So at least it will be relevant for oncologists out there across the country. So this is an example of how the new definition can complicate our interpretation of prior clinical trials. So this is a very well-known trial among in the neuro-oncology community called the CAT-NON trial. And the CAT-NON trial was a randomized trial, a so-called phase 3 trial, and I'll talk about the different phases in a moment, for patients with what were called anaplastic grade 3 gliomas that supposedly did not have this molecular feature of chromosome 1p and chromosome 19q co-deletion. They didn't miss those two pieces of the genetic material in their cells, in the cells of the tumor. So patients that had those tumors with those qualifications, and understandably, this trial did include that molecular feature, but didn't include IDH. So patients in the trial were randomly assigned to either get radiation after surgery or radiation with temozolomide, the chemotherapy agent after surgery. And then in each of those two groups, they were randomized again, and half the patients did not get temozolomide after their radiation or radiation plus temozolomide, and half the patients did get additional temozolomide. And the trial asked two questions. One is giving temozolomide with radiation better than radiation alone? And the second question was, is giving temozolomide after the radiation phase better than not giving temozolomide? And the reason I say is that in the report of this trial, so far we know that giving adjuvant temozolomide was in fact better than not giving adjuvant temozolomide, but we did not see a benefit to concurrent temozolomide, that is giving it with the radiation. However, now that we know the IDH status, and now that we went back and looked at those patients' tumors and looked to see what the IDH or isocitrate dehydrogenase mutational status is, what we found is that indeed in the IDH mutant tumors, there was a small benefit, and perhaps over time this will turn out to be a larger benefit, to giving the radiation and the temozolomide. But this was not seen in the IDH wild-type tumors. So this is a very unusual finding, and one in which if we had had knowledge of IDH at the time this trial was designed and patients were enrolled, it might have allowed us to give a more definitive answer as to what is the best treatment based on IDH status. Another development in the tumor classification is the development of tumor methylation status. So I'm not going to spend time describing what methylation is, but it's a change in the way the DNA of the tumor cells is modified. And by using the methylation status of tumors, it's possible to classify tumors that historically had been difficult to classify simply by looking at them under the microscope. So this very complicated figure from a paper from a couple of years ago, what it shows is little clouds of dots with different colors, and each little cloud of dots represents a different diagnosis. So thousands of patient tumors were submitted, methylation was determined, and then a complex computational algorithm was applied, and it was possible now to assign each of those tumors to the correct diagnostic group. And what I mean by the correct diagnostic group is when you look at this figure here on the right-hand side with all the lines crisscrossing, on the left was the diagnosis that was obtained by looking under the microscope, and on the right is the diagnosis that was obtained by looking by doing the methylation status. And what we learned, in fact, is that some tumors have actually been misdiagnosed based on just looking under the microscope. That looking under the microscope alone is not enough for some tumors to make the correct diagnosis, and that using this methylation classification provides a much greater level of reliability and detail. A few centers, including my own center, have begun doing this for all patients with that come to our center with brain tumor diagnosis. We figure out a more detailed diagnosis using methylation. Okay, I just want to spend a couple of minutes talking about clinical trials, how they are designed, before I go into some detail on clinical trials that are active right now. The first thing I want to say is that overall in the United States, participation in clinical trials by patients with cancer in general, but particularly with brain tumors, is actually very low. Fewer than 15% of patients participate in clinical trials. And there is an understandable hesitancy to do so. So I want to be very honest and say that participation in a clinical trial does not guarantee a better outcome for the patient. We do, however, develop clinical trials with a scientific hypothesis or belief that we think is true. So the goal, of course, is to improve the outcome, but we don't know. And because we don't know, and because we randomize patients, that is by flip of a coin, patients are assigned to either get a standard treatment in many trials or an experimental treatment. We really don't know, or otherwise wouldn't be ethical to put patients in a standard treatment if we knew for a fact that the experimental treatment was actually better. So we don't know, but we have a belief that it might be better. So you say, well, why participate in clinical trials if it's such a sort of a crapshoot, right? We're just flipping a coin. Well, what we do know is that when patients participate in clinical trials, overall, they tend to do better. And that's because of the increased level of care and attention, even for patients getting the standard of care or control arm in that trial. Now, there are many, many trials available, and there's no way in the time that we have that I could go through the hundreds and hundreds of trials that are available around the country and around the world for patients with malignant brain tumors. And I would certainly encourage all of you on this webinar to consult with your local oncologists, as to which trial is best for you, or to seek out a center that may offer additional clinical trials. What I will focus on is some of the more widely available clinical trials, most of which are sponsored by the NCI, which is the National Cancer Institute, which is part of the National Institute of Health. The NCI funds a number of large groups that conduct clinical trials that include collectives of hundreds of hospitals and medical centers around the country. Those groups are called the NRG Oncology is one group. The Alliance for Clinical Trials in Oncology is another group. Another group is the Eastern Cooperative Oncology Group, and there are some other groups as well. And then, of course, the other area you'll find large trials are those that are sponsored by the companies themselves that make the drug or equipment that's being tested in the trial. So I also want to talk briefly about the phases of a clinical trial, because we talk about this often. We'll mention phase 1, phase 2, phase 3. What does that mean? So many of you may have heard that it takes upwards of 15 years from the discovery of a potential target of therapy until the point where the trial has been fully vetted for use with patients. The first part of that, of course, is laboratory studies, the so-called preclinical studies, and often that occurs in universities and academic medical centers. But when it comes to the clinical trials that we talk about, the phases, there are some additional phases, but I want to focus on the phase 1, 2, and 3, and just clarify what their purpose is, because you will often be faced with clinical trial options wherever you are, and it's important to understand the differences. A phase 1 trial is not designed to determine if the drug or treatment is effective. It is designed to determine if the drug or treatment is safe, and at the same time determine what the safe dose or treatment regimen is for that particular thing. So it's very important you understand that if you enroll in a phase 1 trial, the goal of that trial is not to determine if, say, that drug is going to have an impact on improving the outcome from that disease. The goal is all about safety. Now, some phase 1 trials include a group of patients, once they determine the optimal dose, called an expansion cohort, which is really a phase 2 trial. So what is a phase 2 trial? Phase 2 trial is either an expansion cohort or a standalone trial, all on its own, to explore the effectiveness of the treatment. So this is really asking that question, is the treatment effective? So phase 2 is effectiveness. And sometimes phase 2 trials are randomized. That is, there may be more than one group, and you get assigned to one group based on the flip of a coin in a computer. The next phase, the phase 3 trial, is really comparing the standard treatment to the experimental treatment, which has now already been shown to be efficacious in the phase 2 trial, and really validating whether the safety and effectiveness in a much larger group of patients is truly, whether the drug is truly as safe as we thought from the phase 1 trial, and whether the effectiveness is what we thought from the phase 2 trial, all compared to the standard of care. So to give you a sense, in many brain tumor trials of the number of patients in each trial, a phase 1 trial may only include between 10 and 30 patients. A phase 2 trial may include between 30 and 80 patients, whereas a phase 3 trial may include between 200 and 800 patients in those trials, depending on the disease. So they're much larger trials. Some phase 2 trials are connected directly to a phase 3 trial, so we call them phase 2-3 trials, where patients from the phase 2 trial are included in the analysis of the phase 3 trial. And so there are many trials today that use that model. If the phase 3 trial shows what it intended to show, then the FDA will review that and determine if that treatment should be approved for use for that disease. There's also something called a phase 4 trial, in which once the treatment is approved and is being used, typically a company will go and collect data on the patients receiving that treatment as a further validation that it is doing what it is intended to do. So phase 1, 2, and 3 trials. So the other thing I wanted to bring up is where you learn about trials. There are many websites, but the one that every company, academic entity, or NCI-funded group is required to report their trial is called clinicaltrials.gov. So clinicaltrials.gov has every clinical trial there is, and it is a requirement that all these trials be entered there. So moving forward in this talk, every trial that I talk about, I will give you a number called an NCT number, and that number is from this database, clinicaltrials.gov. So if you want to look up the trial, you can simply put the NCT number right here where it says other terms into the search box, and that trial information will come up. Here where it says condition or disease, you might put glioblastoma, brain metastases, and of course there's some other search criteria you can put here in this simple search box, whether it's recruiting, whether all studies means some of the trials may have been finished already, but they're still in the database. If you click advanced search, there are many more search parameters you can enter to help narrow things down, including locations and further details about age and so forth. So that's clinicaltrials.gov. Okay, so let's talk about some of the clinical trials that are ongoing or have recently been completed or completed accruing patients. I'll start with trials for patients with glioblastoma. So this trial called the NRGBN007, and again, here's that NCT number I talked to you about, is asking the question whether adding these immunotherapeutic drugs, ipilimumab and nivolumab, two drugs which have been established for the use in the treatment of melanoma, whether adding those drugs for patients with newly diagnosed glioblastoma, so not recurrent tumors, but new tumors, and that those tumors have to have no methylation of the MGMT promoter. So those of you familiar with glioblastoma know that we always check for promoter methylation of MGMT. This is a gene whose protein product is responsible for resistance to the drug temozolomide. So they have to have unmethylated promoters. That indicates they may be resistant to temozolomide. So in this trial, the patients get ipilimumab and nivolumab with no temozolomide, and they get radiation, of course, or they get standard of care treatment, which is radiation and temozolomide. This trial, NRG-BN001, is almost completed accrual. It asks the question whether giving more radiation dose, so instead of the standard dose, which is 60 gray, the dose is increased to 75 gray. This is randomized, so half the patients get 60 gray and half get 75. It's actually 2 to 1, so for every 2 patients getting 75, 1 patient gets 60. And it includes 2 types of radiation, standard radiation or X-rays or photons, and a kind of particle radiation called protons, which is only available at select centers in the country. The preliminary results of the photon group were reported last year and showed that there was no benefit to increasing the dose to 75 gray. The results of the proton group are not available yet because there are still patients that need to be accrued to this trial. Excuse me. The NRG-BN011, or we say BN11 trial, is looking at patients whose tumors have MGMT methylation, and that means that they're sensitive to temozolomide. And the question is can we improve the outcome of these patients by giving 2 drugs that do similar types of things? So one drug is called temozolomide, of course, and the other is called lomustine. So they combine these 2 drugs. But again, this is a randomized trial, so half the patients will get temozolomide alone and half will get temozolomide and lomustine. And that's only for patients with glioblastoma that have MGMT promoter methylated tumors. The Alliance, which is one of those NCI-funded groups I mentioned, A071102 trial, looks at a drug that's involved in DNA damage repair. So a lot of the treatments we give, like temozolomide and radiation, damage the DNA of the tumor cells. That's how they work. Veliparib blocks the repair of that damage. And so this trial, which has finished accruing patients some time ago and will hopefully be reporting its results in the very near future, added veliparib to temozolomide for patients with newly diagnosed glioblastoma. I'm going to talk in a next slide about this trial, GBM Agile. This is a complex trial that I have a figure to show you because it's hard to explain, but one which may be appealing to many patients out there. And then the last trial I'll just highlight it for glioblastoma is called the Novacure Trident trial, or EF32 trial. This is adding tumor treating fields to radiation. Tumor treating fields added to temozolomide has already been shown to be beneficial, but tumor treating fields added to the radiation component of treatment for glioblastoma has not been demonstrated as beneficial yet, and that's what this trial is asking. A couple of other trials just worth mentioning. I already talked about the Catnon trial, but here is the NCT number, and I mentioned the results of this, at least in early form, have been reported now and are in publication, and I mentioned those. A similar trial, in this case, similar types of tumors, but this case they have co-deletion of those two chromosomes I mentioned, 1p and 19q. That's called the CODEL trial because of co-deletion, and that trial is still ongoing and accrual, and we don't have results yet. For patients with tumors who are IDH mutant, and of course now we would say that does not include any glioblastomas, this trial, NRG-BN005, compares standard X-rays or photons to particle therapy or protons, the same dose, but just different types of therapy, and looks to see if it can improve neurocognition or decrease the neurocognitive effects of radiation. For meningiomas, the Alliance A07-1401 is looking at various targeted therapies that target different genetic changes in different subsets of tumors and for progressive disease. And this is an ongoing trial. And another ongoing trial is the NRG-BN003. And this is asking the question is for patients that have grade 2 tumors. So meningiomas only have 3 grades, grade 1, 2, and 3. For those in the middle, the grade 2 tumors, is it better to just watch and see if the tumor grows back and treat then, or is it better to give radiation after surgery to remove the tumor to prevent it from coming back? And that's a question we don't have the answer for yet. That's an ongoing trial. So I mentioned the GBM Agile trial, and this is a really unique trial run by a new organization called the Global Coalition for Adaptive Research. It's not funded by the government, and it's an adaptive platform. And this is very complicated. I'm just going to kind of walk you through it very quickly. The idea of this trial is that we can continuously add new drugs and treatments and test them without creating a new trial from scratch. So there are, as patients enter into this trial, they get randomly assigned to whatever treatments are currently available in the trial. Currently, I think there are 4, and a 5th is being added, as well as the standard of care. Now, for the standard of care group, once that group has finished, the predefined number of patients has been enrolled, then we don't need to keep enrolling patients in that group. And then the other groups get enrolled. With each new patient, the outcome of the previous patients is analyzed. And a decision is made whether that therapy is even effective. And if it isn't effective, there's some predefined point where it says this is futile, this particular treatment that's in this larger trial is not effective, then that group of patients is no longer increased. In other words, patients are no longer assigned to get that therapy, which is deemed ineffective. Now, on the other hand, if that therapy is found to be effective with each new patient and it reaches a certain threshold, then it's possible to now declare that is an effective treatment and either send it on for a further trial, or at the very beginning of the inclusion of that therapy in the trial, it can be pre-specified that an additional group of patients, say 50 patients, will get that therapy if it reaches that threshold of showing effectiveness. And once it has done that is enough. That then means that the drug is deemed effective and the FDA can review it for approval. The benefit of this is it takes far, far fewer patients enrolled to get to the point where you have approval by the FDA, far, far fewer. And you don't have to keep creating new trials because each new trial creation normally takes several years to develop. This is a new treatment regimen can be added to this scheme really in the matter of months. So it's quite an effective method. So many centers now have this trial running, and it's available both for patients with newly diagnosed glioblastoma as well as recurrent glioblastoma. So as I said, adaptive trial benefits is that there are fewer patients required to answer the question. Only one control group is needed as opposed to creating a new trial with a new control group with each new trial. And that can be continuously modified as new therapeutic questions are asked. New drugs come out, new drug combinations can be added immediately. And this can quickly lead to approval by the FDA as opposed to waiting 4 or 5 years for every trial before learning whether it can be approved by the FDA. I also want to emphasize how there are different treatments for patients with glioblastoma based on the MGMT status. So I mentioned it briefly. The MGMT gene encodes for this protein called O6-guanine DNA methyltransferase, which is involved in the repair of damage from so-called alkylating chemotherapy. So drugs like temozolomide and lomustine are alkylating chemotherapy. That's how they damage DNA, and this gene repairs them. So modification or methylation of the regulatory region of the gene reflects the ability to resist chemotherapy treatment, but it's also associated with overall outcome. So MGMT unmethylated tumors are found to be more resistant to alkylating chemotherapy like temozolomide or lomustine, but it also may reflect a higher risk group. And in higher risk groups, we're more likely to offer novel therapies. So in the case of the NRG-BN007 trial, we're offering immunotherapy in place of temozolomide because that's a high-risk group of patients. MGMT methylated tumors may be more sensitive to temozolomide. So in that case, if they're sensitive to alkylating chemotherapy like temozolomide, why not give 2 alkylating chemotherapies? And indeed, an earlier trial, a phase 2 trial, showed that this might have a benefit. It was a trial conducted in Europe. Okay, let's talk a little bit about immunotherapy. Primary brain tumors treated with immunotherapy is really the focus of hundreds of clinical trials available right now. None has yet reached the point of FDA approval. Some of the approaches that are being used in these trials, the most common and earliest trials, try to stimulate the production of tumor killing cells. These are the effector T cells, they're sometimes called. And these drugs were called checkpoint inhibitors, and they came from successes in melanoma and in lung cancer, drugs like ipilimumab and nivolumab. Another class of treatments is chimeric antigen receptor modified tumor killing cells, CAR T cells, in which case the T cells are engineered to target a particular protein that is found on the tumor cells. There are many vaccine trials that use specific mutant tumor proteins, either known to be present in a variety of tumors or derived from the patient's own tumors, sort of personalized vaccines in that case. And there are trials that have engineered viruses that attack both the tumor cells, but also stimulate the immune system to attack the tumor cells. So what I mean by that, stimulating the immune system, is when you get a cold, that your immune system is activated and you build up a huge immune response. Of course, that gives you the congestion. I don't have a cold right now. My voice is my throat is just a little dry from speaking, but that is what viruses do. And so what has been found is that some of these viruses in these trials actually cause an inflammatory response. And in that inflammatory response, the immune system also attacks not only the virus, but the cancer cells around the virus. So one study that came out a couple of years ago that showed some promise with checkpoint inhibitors was this trial, which used another checkpoint drug called pembrolizumab, which is similar to that nivolumab, for patients with recurrent glioblastoma that were going to have a second surgery. And what they found is that if they gave the drug before the surgery for a while and then did the surgery, that these patients did better than giving the drug after the surgery. And what this taught us is that there might be a benefit to having tumor in place when you give this kind of drug, as opposed to doing it when all the tumor that's visible has been removed. And you can see here that this was the tumor after initial treatment, and that was some months later. There was evidence that the tumor was growing and progressing, and so the patient was seen just before surgery. They had surgery, and they were given pembrolizumab, and then after treatment, the tumor came back. So this is kind of how these tumors are defined, and this is a case where they gave the pembrolizumab after surgery, and as I said, in this case, the tumor came back, whereas if they gave it before surgery, there was a clear benefit. This was a small trial with a few number of patients, but but it gives some suggestion that there might be a benefit to treating this way with the tumor intact. Okay, I know there was a lot on primary brain tumors, and I do want to switch gears to make sure I cover the treatment of brain metastases. So what do we mean by brain metastases? So again, these are tumors that develop from cancers elsewhere in the body. The most common are lung cancers, melanoma, and breast cancers. And there are other types, and melanoma, kidney cancers, even rarely cancers of the GI tract like gallbladder cancer. Those tumor types also tend to show intratumoral bleeding. They bleed inside the tumor. Up to 30% of cancer patients develop brain metastases at some point during the course of their disease. And at least 200,000 patients annually are diagnosed with brain metastases. Now, I don't know if you remember the slide I showed you on primary brain tumors, but that's 10 times more than the number of patients that are diagnosed with primary brain tumors. So it's a very large number. It's more, for example, than all the patients that are diagnosed with lung cancer every year or with breast cancer every year. So brain metastases is a major problem. The key therapeutic approaches that are used to treat patients with brain metastases are surgery, and that removes large tumors or tumors that are isolated. They only have 1, 2 tumors. And radiation, which is used for unresectable tumors or when multiple tumors are present. And in fact, we use radiation almost always after removal of a large tumor. We radiate the empty space, the tumor cavity, to make sure the tumor doesn't come back. So this is a very big field. And again, just as I said in the beginning, it's hard for me to cover everything. But I want to focus on a couple of the key questions that are under investigation for patients. One is, how do we minimize toxicity? There's a great concern about the toxicity from our treatment. Our goal, of course, is not to make patients feel worse, but to make them feel better, particularly cognitive changes, which can be associated with both chemotherapy and radiation therapy. Can some of these treatments have an effect outside the brain as well as in the brain? When is it beneficial to treat the whole brain with radiation as opposed to just the individual tumors? And what is the best sequence for giving drug treatments and radiation, particularly for patients that have cancers where immunotherapy is used, like melanoma and lung cancer? So let's define the different treatments that we have. This is whole brain radiation. So this is what looks like an X-ray of a patient standing sideways. So this is the head here. And those blue lines over the face, I like to emphasize to my patients, are preventing radiation from going to the face. So when we give whole brain radiation, we're really just radiating the brain. But it is given to the entire brain, and it takes 2 to 3 weeks to give. Another type of treatment you may have heard of is called stereotactic brain radiation, sometimes called stereotactic radiosurgery. This is radiation only to the tumor that we want to treat, not to the whole brain, and it's usually given in 1 session, or sometimes stereotactic radiotherapy is given in 3 or even 5 sessions. Here's an example of a machine used to treat. This is a gamma knife. This is one of the many types of ways to give stereotactic brain treatments. So when we treat with whole brain radiation, one of the most important studies that's been out now for more than a few years was this study called the RTOG 0614. The RTOG was the predecessor to the NRG, if that all these letters I know are hard to remember. But in this trial, what we found is that giving a drug called memantine, which is a drug actually used to prevent memory decline in patients with Alzheimer's dementia, also prevented memory decline in patients who got whole brain radiation. And as a result, for most radiation oncologists, it is now standard to give memantine to any patient getting whole brain radiation. And one of the things I wanted to state is that I typically give this continuously. I don't stop it after a couple of months. Some of the confusion you might hear about is the clinical trial did stop the treatment after a couple of months, after 6 months, but that was not because the treatment wasn't beneficial. That's because in a clinical trial, you have to stop the treatment at some point in order to see what its effectiveness is. So they had to have a predefined time. But in practice, there's no reason to stop. It's a very well tolerated drug, very inexpensive drug. One way when giving whole brain radiation to avoid cognitive effects is actually to avoid treating the sensitive, memory-sensitive areas of the brain. In this case, this area called the hippocampus. So this is the same sort of sideways view, zoomed in a little bit. This is the eye over here. And this is the hippocampus. It's supposed to look like a seahorse, but I never think it does. And hippocampus is the Greek word for seahorse. And if we avoid giving radiation, which is being done here, it has been shown that there is improvement or a decrease in the neurocognitive decline associated with whole brain radiation. And recently, just a year and a half ago, this trial, the NRG-CC001 trial, combined memantine with that avoidance of the hippocampus and showed a further benefit. So when given whole brain radiation whenever possible, we try to combine the two approaches. Now, one of the challenges of doing that, though, I'll talk about in a few minutes. Another way to decrease effects to the normal brain, including cognitive effects, is to use stereotactic treatment. As I mentioned, this is highly focused. It does not really treat surrounding brain tissue, just the tumors. And increasingly, this is being used for large numbers of tumors. Historically, after 3 or 4 tumors, we would do whole brain radiation, but now things have changed. And it's even being applied to tumors that previously were considered poor candidates for stereotactic treatment. So here's a large study where stereotactic treatment was used for a kind of lung cancer called small cell lung cancer, where typically we only did whole brain radiation, and it showed that, in fact, patients did very well and even better with radiosurgery first, stereotactic treatment first. So here are some trials in which larger numbers of tumors are being treated with stereotactic radiation. Two trials at MD Anderson, and I should disclose that I used to work there for many years, are looking at large numbers of tumors. A trial in the Netherlands looking at quality of life outcomes for patients getting treated to upwards of 10 tumors, and a very large trial done by the Alliance, as well as a group in Canada, looking up to 15 tumors, comparing whole brain radiation with hippocampal avoidance to stereotactic treatment. So can therapies be used to treat tumors that we use outside of the brain? Can those be used to treat tumors in the brain? And a few years ago, several trials were tested in melanoma where they looked at brain metastases, and they found that there was a response in the brain metastases. They shrunk when given with treatments that are historically used for patients with melanoma. This includes immunotherapies like nivolumab and ipilimumab, and so-called targeted therapies that target specific mutations in melanomas called the mutation of the BRAF gene, and those drugs are called dabrafenib and trametinib. And all of those showed a response in the brain, suggesting they would be beneficial for patients with brain metastases. Indeed, there's an ongoing trial by the Alliance called the A071701 trial. And again, here's that NCT number. And the idea is to look at the tumors, if available, a tumor that's been resected from the brain, if not that, the tumor that originally is in the patient that metastasized to the brain. And based on these particular genetic changes, and I won't go through each of these genes, a different drug is given that targets these genetic changes in the pathways associated with them. So this is an ongoing trial. It's a very interesting approach. It is a phase 2 trial. So again, looking at efficacy. It's important to note, though, that trials that look at radiation often use different endpoints than trials looking at drugs. And so sometimes we're comparing drugs to radiation, and it's a bit of an apples and oranges kind of comparison. In general, radiation trials look at functional changes to the patient, such as neurocognition. as opposed to just changes to the tumor size on an image. As it turns out, changes to the diameter of a tumor on an image really doesn't have any good correlation to how a person is doing, how they're functioning. And so radiation trials tend not to be as focused on that as drug trials. So sometimes it's hard to make the comparison. I think the best approach is really to combine treatments and to have a very open discussion with your caregiver, with your care provider, with your physicians, as to what is the best order of treatment. Is it better to give the drug first or give the radiation first? Stereotactic treatments are often very amenable to sort of give the treatment and start the drug the next day, or even give the treatment while the patient is receiving the drug. Whole brain radiation often requires stopping the drug for several weeks, so sometimes that decision about sequencing comes into play. So I'm going to briefly talk about some of the new technologies that are out there that are hopefully going to be of interest in the future. One question that's been asked recently is, can we visualize tumors better than we've done in the past? The typical MRI that's been done for many, many years to visualize tumors is in fact not great in distinguishing between tumor growth and treatment effects on the tumor that don't reflect tumor growth. So these are some agents that are being tested. One is this drug, fluorine 18 flucyclovone, which is a drug that you include in a PET scan, and it seems to do a good job of detecting active brain metastases. And so there's some ongoing trials looking at this agent in patients with brain metastases as well as with primary infiltrating gliomas. Another drug called gadolinium-68 dotatate is looking at meningiomas. It binds to a receptor protein on the surface of meningioma cells, and again, in a PET scan, is very good at visualizing meningiomas. A variation of this drug where the gadolinium-68, which is a radioactive material, is replaced with a more powerful radioactive material, is being used to treat meningiomas, and I have to disclose I am the investigator on that trial. We can also try to increase the dose to the tumor bed. This is a treatment that uses a radioactive compound called cesium-137 embedded in these little stamp size pads that go into the tumor cavity and can be used to deliver a very high dose of radiation just around the tumor cavity, but not to the normal brain. It's referred to by that brand name. And new ways to deliver stereotactic radiation. So the Gamma knife, which has been around for 50 years, has been upgraded time after time and now has a system called the ICON system, which includes the ability to do a quick CAT scan while the patient is on the table to allow for treatments without the use of what's called a stereotactic frame. Another type of radiosurgery treatment or stereotactic treatment is uses a treatment based where the patient is inside this chamber with a gyroscope to help line them up. So it's another way to deliver stereotactic treatment. There's novel approaches for treating when the cancer has spread outside the brain tissue into the fluid that lines the brain, the so-called leptomeningeal disease or leptomeningeal carcinomatosis. And so there's a recent trial that was reporting using proton therapy to treat the brain and spinal cord where that fluid surrounds with some effect. And there's also approaches giving immunotherapy inside this fluid directly that are being explored right now. Okay, so to summarize, there have been significant changes in the way we classify primary brain tumor that really have an impact on the way we interpret past and ongoing clinical trials and how they guide the best treatment for patients. Multiple approaches are currently under investigation for the treatment of brain tumors, and there are some trials that are going to report in the next several months that I would keep an eye out for. Patient functioning remains critical, and I have not been able to give full justice to all the work that's being done focusing on improving patient quality of life and patient functioning. I talked a little bit about neurocognition and the studies that are being done on brain metastases. Rational approaches to combine treatments with drug treatments and radiation treatments are leading to improvements in outcomes for patients with brain metastases. And of course, emerging technologies will further improve function and outcomes. So I want to thank you very much for listening to this, and we'll see if we can get some of the questions answered.
Thank you so much, Dr. Sulman. As a reminder, if you have questions that you'd like to ask, please type and submit it using the Q&A button on the bottom of your screen, and we'll answer questions as time allows. So Dr. Sulman, one of the first questions we have here is regarding treatment for brain metastases. Is brachytherapy something that is available for patients experiencing brain metastases?
So there is the slide I showed you on emerging technologies with the cesium implants is a brachytherapy, indeed, and it is being tested for patients with brain metastases. So indeed, that is there are some variations on that theme, but that is indeed what is being done.
Thank you. And what about patients who are on wait and watch? Can they qualify or be eligible for any of these treatments that you've discussed? And if so, how do you have any advice on how they can bring this up with their treatment team?
Right. So, there are some types of infiltrating tumors, for example, where the approach maybe after surgery or even without surgery is to observe obtaining imaging at a regular interval. So the so-called watchful waiting. And it is worth having the discussion even in those follow-up visits, even when you've kind of come to the consensus that we're not going to do anything now, we're going to wait, to say, look, let's talk about some of the treatments that will be available when and if it's necessary to treat that tumor. So I would definitely bring it up with your oncologist, and you're welcome to talk about the relevant trials that I mentioned. Absolutely.
Thank you so much for that insight. During your presentation, you shared the great resource of clinicaltrials.gov, which can be really helpful when researching clinical trials on your own. It can be a little tricky to use. Do you have any advice or tips on patients and families who are using this resource on how to narrow down trials and find ones that may be applicable for them?
I agree. It is difficult to use. My suggestion, and one that I encourage my patients to do, is ask that your physician do it with you in the office, and do it together, because it is technical often, and it is. I like to say it's designed poorly both for patients and for physicians. So nobody wins with that design, and so it's better to do it together. And I would definitely encourage you to do so.
That's wonderful advice. Thank you so much for sharing that. I think we might have time for maybe one or two more questions. You talked about participation in clinical trials and how quite a few patients actually participate. Do you know if COVID has impacted this at all?
It has, and unfortunately, during the past 2.5 years, trial participation declined dramatically, and understandably so. It was patients were reluctant to come in as often as necessary for the visits associated with clinical trials. There was a legitimate fear of being in a major medical center in a major city. I'm in New York, and there was many of my patients were terrified to come into New York City during that time. So there was a significant drop-off in clinical trial participation. We have seen a very good rebound, but we still are nowhere near the levels we'd like to see for clinical trial participation. And I hope I've conveyed to this group and to this audience that there is a benefit for participating in a clinical trial, regardless of what the question of the trial is asking, just by being in a trial.
Thank you so much, Dr. Sulman. Unfortunately, that is all the time that we have for questions. Thank you once again, Dr. Sulman, for your wonderful webinar presentation and for your time today.
And I just want to, you're very welcome, and thanks you to everyone. And I did say that I'm going to delay my next meeting a few minutes. I'll try to answer a few of these typed in if I can, because I know people asked a lot of questions. I'll do my best to respond.
That would be wonderful. Thank you so much for offering that.
Okay.
All right. Well, Thank you again, Dr. Sulman. Additionally, I want to thank our webinar sponsor today, GT Medical Technologies, for their support of this program. And most importantly, I want to thank each and every one of you for joining us online and sharing your questions. We hope that you found some benefit from this webinar and some information that you might not have known before. Aside from our educational webinar series, the ABTA has a variety of programs that help support patients and caregivers throughout their experience. For more information about ABTA's programs, events, and services, visit ABTA's website at abta.org, email us at info@abta.org, or call the ABTA Care Line at 1-800-886-2282.